Imagine a world where diseases like cystic fibrosis, sickle cell anemia, and Huntington's disease are a distant memory. This is the promise of gene editing therapy, a revolutionary approach to medicine that is rapidly moving from the realm of science fiction to reality. Scientists around the world are already using gene editing tools to develop groundbreaking therapies, offering hope to millions affected by debilitating and life-threatening illnesses. As we stand on the cusp of this medical revolution, it is crucial to understand the potential benefits and challenges of gene editing, ensuring that this powerful technology is used responsibly for the betterment of humanity. The future of medicine is being rewritten, and gene editing is at the forefront of this transformation. Sarah and David were overjoyed at the birth of their daughter, Emily. However, their joy soon turned to fear when Emily was diagnosed with cystic fibrosis, a genetic disease that affects the lungs and digestive system. The doctor explained that Emily had inherited a faulty gene from both parents, causing her body to produce thick, sticky mucus that clogs her airways and makes it difficult to breathe. The news was devastating. Sarah and David knew that cystic fibrosis is a progressive, life-shortening disease with no known cure. One day, they came across an article about a new experimental therapy called gene editing. This revolutionary approach, the article explained, could potentially correct the faulty gene responsible for Emily's condition. Emily's story highlights the profound hope this technology offers, while also underscoring the importance of carefully considering its ethical implications. At its core, gene editing is a way to make precise changes to an organism's DNA. Think of DNA as a long instruction manual that contains the genetic blueprint for building and maintaining a living organism. Gene editing tools act like molecular scissors, allowing scientists to cut DNA at specific locations. By introducing new genetic material during this repair process, scientists can essentially rewrite the instructions in the DNA, correcting genetic defects or introducing beneficial traits. This ability to precisely target and modify genes has the potential to revolutionize medicine. One of the most powerful and widely used gene editing tools is called CRISPR-Cas9, often shortened to CRISPR. CRISPR is a naturally occurring system in bacteria that acts as an immune defense mechanism against viruses. Scientists have harnessed this system to create a programmable gene editing tool. Here's how CRISPR works. A short RNA molecule called guide RNA is designed to match the sequence of the target gene that needs to be edited. This guide RNA acts like a GPS, guiding the CRISPR system to the precise location in the DNA. The guide RNA is attached to a protein called Cas9, which acts as the molecular scissors. Once the guide RNA finds its matching sequence in the DNA, Cas9 cuts both strands of the DNA molecule at that specific location. After the DNA is cut, the cell's natural repair mechanisms kick in to fix the break. Non-homologous end-joining NHEJ is a quick repair mechanism that often introduces small insertions or deletions of genetic material at the cut site. Homology-directed repair, HDR, is a more precise repair mechanism that uses a template DNA molecule to guide the repair process. CRISPR's ability to precisely target and modify genes has revolutionized genetic research and opened up unprecedented possibilities for gene editing therapy. Gene editing therapy is already making significant strides in treating a range of diseases, offering new hope to patients who previously had limited treatment options. Blood disorders. Clinical trials are underway using CRISPR to treat sickle cell anemia and beta thalassemia, inherited blood disorders that affect the production of hemoglobin. Cancer. Gene editing is being explored as a potential cancer treatment by modifying immune cells called T cells to recognize and attack cancer cells more effectively. Inherited blindness. Researchers are investigating the use of CRISPR to correct genetic mutations that cause inherited blindness, such as Leber congenital amaurosis. These are just a few examples of the many ways gene editing is being used to develop innovative therapies. As research progresses, we can expect to see even more applications of this powerful technology in the fight against human disease. The potential of gene editing to transform medicine is undeniable. However, we must ensure that this revolutionary technology benefits all members of society, not just a privileged few. Looking ahead, the potential applications of gene editing therapy are vast and awe-inspiring. Scientists envision a future where gene editing could eradicate inherited diseases. By correcting faulty genes in embryos or germ cells, we could potentially prevent inherited diseases from being passed down to future generations. Treat complex diseases. Many common diseases, such as heart disease, Alzheimer's disease, and diabetes, are influenced by a combination of genetic and environmental factors. Gene editing could be used to target multiple genes involved in these complex diseases, offering more effective treatments and preventive strategies. Enhance human capabilities. While still in the realm of science fiction, some speculate that gene editing could one day be used to enhance human traits such as muscle mass, intelligence, or longevity. The possibilities of gene editing are both exciting and daunting. As we continue to unlock the secrets of our genetic code, we must proceed with caution and carefully consider the ethical implications of this powerful technology. While gene editing holds immense promise for improving human health, it also raises profound ethical considerations that must be carefully addressed. Safety is paramount. As with any new medical technology, gene editing tools are still under development, and there is a risk of unintended consequences, such as off-target edits or unforeseen long-term effects. Rigorous research and clinical trials are essential to ensure the safety and efficacy of gene editing therapies. Informed consent is also crucial. Patients undergoing gene editing therapy must be fully informed about the potential risks and benefits of the procedure. Justice and equity are other significant concerns. Gene editing therapies are likely to be expensive, at least initially. It is crucial to ensure that these life-changing therapies are accessible to all who need them, regardless of their socioeconomic status. Navigating these ethical complexities requires open and transparent dialogue among scientists, ethicists, policymakers, and the public. The potential of gene editing therapy to transform medicine is undeniable. However, we must ensure that this revolutionary technology benefits all members of society, not just a privileged few. The development and implementation of gene editing therapies are likely to be expensive, at least in the initial stages. This raises concerns about equitable access, as these life-changing treatments may only be accessible to those who can afford them. To prevent this scenario, we must prioritize equitable access to gene editing therapies. This requires affordable treatments by exploring innovative funding models, such as gene editing, specific insurance plans, or government subsidies. Global collaboration is also key, sharing research findings and resources internationally to ensure that gene editing benefits people in all parts of the world. By prioritizing equity and accessibility, we can harness this transformative technology to create a healthier and more just world for everyone. One of the most ethically charged aspects of gene editing is the possibility of germline editing, which involves modifying the genes in sperm, eggs, or embryos. Unlike somatic cell editing, which targets only the cells of the individual undergoing treatment, germline editing produces changes that are passed down to future generations. The prospect of germline editing raises profound ethical questions about our responsibility to future generations. Proponents of germline editing argue that it has the potential to eradicate devastating genetic diseases, sparing future generations from suffering. Opponents argue that germline editing crosses a fundamental ethical line by allowing us to play God with the human genome. They worry about the potential for unintended consequences, such as unforeseen mutations or off-target effects that could have harmful consequences for future generations. The debate over germline editing is likely to continue for years to come. It is a complex issue with far-reaching implications that requires careful consideration and open dialogue among scientists, ethicists, policymakers, and the public. Gene editing has the power to revolutionize medicine and transform human health in unprecedented ways. As we stand on the cusp of this new era, we must proceed with both excitement and caution, embracing the potential benefits of this technology while carefully considering its ethical implications. Shaping the future of gene editing requires a collective effort, a global conversation that includes scientists, ethicists, policymakers, patients, and the public. We must work together to ensure that this powerful technology is used responsibly, ethically, and equitably for the benefit of all humankind. Stay informed by educating ourselves about gene editing, its potential benefits, and its ethical considerations. Engage in open and respectful dialogue with others about this complex issue. Support responsible research by advocating for continued funding for gene editing research and clinical trials, while ensuring that ethical guidelines are followed. By working together, we can harness this transformative technology to create a healthier and more equitable world for ourselves and for generations to come.